this video, I'll be going through some last-minute tips to help you guys ace the ACP paper. This is for 610 Biology IGCSE for the CIE spec, but I think it may be applicable to other papers as well. So there are a lot of things that could come up in this paper, as it's a practical paper, and I've split this into 3 categories, so we can split the things that come up in this paper into experimental design skills, handling data, and practical knowledge. So the things that come under experimental design are identifying variables, for example, your independent, dependent, and control variables, suggesting methods and safety precautions, and also proposing ways to improve the reliability of an experiment and to reduce any errors. Handling data is just the mathematical side of this paper, so it's just doing calculations like averages, percentage change, and magnification, and also being able to spot anomalies in data and just identifying the general trends given data from graphs, tables, and charts. And practical knowledge is the actual, like the experimental things like your food tests, microscopy, and the experiments that you would have come across in the course, for example, enzymes, photosynthesis, respiration. Anyway, there are 3 types of variables: independent, which is what we change, dependent, which is what we measure, and control, which is what we keep the same. Normally in an experiment we have multiple control variables, but only one independent variable and one dependent variable, so keep that in mind. So there are experimental precautions that we have to follow, precautions that we must carry out essentially, such as for liquids, taking liquid readings at eye level with the eye perpendicular to the scale, and this is to prevent parallax error, and take the liquid readings from the bottom of the meniscus. As usual, for all experiments we must take repeat readings and average unless this is impossible. Use a heat shield to prevent heat from a light source affecting an experiment. This is specific to just the photosynthesis experiment, but it's just an example of one of the many precautions that could be taken just to avoid external or other factors from affecting the results of an experiment. We can stir solutions so that the components of a solution are evenly distributed, and use burettes and pipettes instead of measuring cylinders, as they're more accurate. There are also safety precautions that we must follow, that we must carry out when doing an experiment, for the safety of the people doing the experiment and also others as well. So stand up while doing an experiment, don't be sitting down with chemicals, don't do that. Wear goggles to protect your eyes. Use tongs to hold the hot test tubes, don't just go touching them with your bare hands. Do not heat alcohols or volatile reagents directly over a flame, use a water bath instead, and when cutting materials, cut away from your fingers. Magnification calculations are something that comes up in every paper every year, so you must know this. So this is the equation for magnification. Magnification is image size over actual size, and one thing I'm going to note is that when calculating magnification is that you must make sure that the image size and the actual size are in the same units. They like to catch people out and give them both in different units, and the unit conversions are not given, so you must know that there are 1,000 micrometers in a millimeter and 1,000 millimeters in a meter. And just like they can ask magnification, you can also calculate things like the image size and the actual size given this equation. You will actually not be given the equation, you must learn the equation. You can use the triangle if you want. If equation rearrangement is something that you don't like, just write I am in a triangle and cover with your finger the thing that you're looking for. So if you want to calculate actual size, cover the A and you're left with an I over an M, which means that actual size is equal to image size over the magnification. So that's how you do magnification calculations. Make sure to do like a final sanity check on your magnification, make sure you are getting a reasonable number, not something like 0.5 times. Magnifications are usually in the thousands because you are zooming in on an object. Scientific drawing. Here are some general rules for scientific drawing. Scientific drawings are not done like artistic ones, so No shading, no feathering, no little artistic things that would not be permitted in a scientific drawing. So make sure that your drawing is large and it takes up most of the space that is provided. Use a sharp pencil, so no pens. Use a clear, continuous line with no breaks and no feathering. So also do not shade in any way. So no like stippling or hatching, any kind of shading. Just leave it blank, okay? And draw exactly what you observe and nothing more. So just because you think something should be biologically present in a drawing, don't add it in because you think it should be there. Literally just copy the reference image, copy that outline. There is a mark just for having like the general shape of the reference image. Here are some notes about labeling your scientific drawings as well. Your label should be written horizontally and make sure your label lines don't cross or intersect, and do not use arrowheads. Literally a horizontal line that points to the thing that you are labeling is perfectly fine. So here are two examples of scientific drawings. The one on the left is significantly better than the one on the right. I'm going to explain to you the mistakes on the one on the right. So you can see here there are a lot of problems. These lines are feathered instead of being a clear continuous line, so that's what they've done wrong here. This is shaded, that should not be shaded. There are arrowheads, so this is like what you should not be doing. As for this one, you can see that they have followed the general shape of the leaf. Like they followed the general outline of this, and there is a mark for doing this. Both diagrams take up most of the space they were given, so that's good. But don't do something like this. So make sure your lines don't cross at any point. This is what you should be aiming for. Now, when drawing graphs, remember the mnemonic SLAPOK, which is just, it stands for scale, line, axes, plot, units, and key. So the scale means that the graph should take up at least half of the space that they're giving you. So don't just draw like a tiny graph, expect to get away with it. Don't do that. The L stands for line. So draw your line of best fit with a ruler. It may not always be a line of best fit, it could also be a curve of best fit. Just whatever you do not connect the points like a dot to dot. Make sure it's a line or a curve which shows the general trend of the data. The A stands for axes, which must be labeled, and also the correct way round. So independent variable on the x-axis and dependent on the y-axis. The P stands for plot your points accurately. Do so with a pencil, not a pen. And U is for units. So don't just leave your axes without units. If one of the axes is length, for example, make sure it's length in meters, length in millimeters, for example. Don't just write length and leave it there. The K is for key, which is not really applicable to every graph, but if you have multiple lines on one graph, then they need to be clearly labeled. So yeah, the key is not always necessary for every graph, but use it when it's appropriate. You must know how to test for the 5 following substances: reducing sugars, proteins, lipids, starch, and vitamin C. Now, for reducing sugars, use Benedict's solution. Heat for a few minutes, and the positive result is a brick red precipitate, and the negative result is the solution remaining blue. This is a semi-quantitative test, which means that the color depends on the concentration. So as you can see, as we increase the glucose concentration, the color will change gradually. It will go from blue to green to yellow to orange to finally brick red. Now, just like they can ask you how to carry out a Benedict's test, they can also ask you things like the hazards and the precautions associated with doing so. So the hazard would be the hot water or the flame, and the precaution would be to stand up during this practical, use a water bath, and use tongs instead of just touching the test tube with your bare hands and burning yourself. The food test for proteins, we use Biarritz reagent. The positive result for this one is a color change from blue to purple, and the negative result is the solution just remaining blue. Now, for lipids, use the ethanol emulsion test. So you add the sample to a few drops of ethanol and shake vigorously. The positive result is something called an emulsion. It's just a layer on the top that is milky white. So look for a milky white emulsion that is being produced. And a negative result is just the mixture remaining clear, so there will be no emulsion produced. For vitamin C, use something called DCPIP. the dark blue DCPIP will decolorize in the presence of vitamin C. So that is it for food tests. This will inevitably come up because you are always asked to plan an investigation for 6 marks. Now there are a lot of mnemonics you can use for this. In this video I'm using IMARCs, which is independent variable. So what are we changing? Independent variable is the thing that we change. It's what affects the dependent variable. Now when we change the independent variable, how do we intend on changing it? Make sure you write that in your method. M is for measure, so we measure the dependent variable, but how? Using which equipment? The A is for accuracy. Are there any sources of error? And how do we mitigate any errors? How would we make sure our measurements are accurate? R is for repeat, so repeat 6 times and take an average. You're bound to get a mark just for saying this. C is for control variables. Which variables do we control and how? State at least 2 or 3. Don't just give one, give multiple. The S is for safety, so are there any hazards? What precaution will we take? Are we dealing with any volatile substances? Are we using any toxic substances? What precautions will we take to mitigate any hazards? So here are some general tips for this paper. Do previous ATP papers, especially the drawing and calculation questions. These come up every time and they often catch people out, so make sure you have some practice at drawing and also at the various types of calculations that could come up in this paper. Make sure you do them under time conditions, and to also go over the experiments that you've done or learned about. Just identify the dependent, independent, and control variables and go through the methods and make sure you understand why things are being done. This is quite important. And when doing the exam itself, just be careful, to read and highlight the keywords like precaution, variable, and control. Underline any keywords. You can annotate the question. Hopefully this video has explained something about the ATP paper. If it's been of any use to you or if you have any questions, do drop them in the comments section. Hopefully this has been of some use to you, so thank you for watching.